Good morning, everybody. I hope you're all well this morning and looking forward to this talk. Without any further ado then, I'm really delighted to introduce Giles. As I said, I think for many people he doesn't need any introduction. We've known him for his numbers of years. Was it 12 years at the NBU? Yeah.
No idea.
Certainly a long time. And you're now up at Newcastle University working on crop and bee health. And as well as being an independent science advisor to the Defra Bee Health Forum. So I actually think that the audience today reflects that, the respect for your knowledge there, Giles. You're recently working on European foulbrood and also trying to get a better understanding of chronic bee paralysis virus as well. So I think we're all waiting. You don't want to hear me anymore. Over to you, Giles.
Thanks a lot for that lovely introduction. Good morning, everybody. Thanks for choosing this lecture over what else is going on at the moment. Great to see so many people here. Today we'll be talking about chronic bee paralysis, which is the disease, not to be confused with chronic bee paralysis virus, which is the causative agent of chronic bee paralysis. So first off, a little bit of audience participation. Those of you who've seen me before will know that I like to do this a little bit just to keep you on your toes. How many people in the room think they may have seen chronic bee paralysis? If you just raise your hand. Wow. That's a good number. Okay. So we'll go through the symptoms and we'll see whether or not that number diminishes or increases. Okay. Great. So I'm up at Newcastle University now. I'm really lucky. I work in a modelling evidence and policy group, which is great because there's people in there that are really good at taking very complex situations and trying to use various different modelling and mathematical approaches to understand them better, to pick them apart, sometimes to simulate them, and then we can think about interventions. And if you think about honeybees and you think about the diseases that they carry and how honeybees interact within a hive and how beekeepers interact at landscape scales quite often, then you can see that they're quite complex biological problems. So it's great to be within this particular team at Newcastle. I've been up there for about 5 years now. We've built a bit of a bee team. We've got lots of sponsorship, which we're very glad of and thankful for over the last few years. And I'll be talking a little bit about some of the work of the folk that are on this screen, and I'll introduce you to them as we go through their work. Last important members of our team are the bees. We've got 2 research apiaries. This is the day that it was set up at Cocker Park Farm, which is a university-owned farm just north of Newcastle. And then the other one is on the roof of one of our buildings at Newcastle University. A terrible place to keep bees. If you're a beekeeper, I wouldn't recommend sticking it on top of a 5-storey building, especially when the storms come rolling in. But what it's great for is the colonies that do thrive through the summer. We can take the bees directly from here and we can take them downstairs, just 1 story, and we're straight into a clean lab where we can handle the bees. And that's really useful for us to be able to be that close to the bees. Going up to the farm to get them would be a real pain. So they're really important members of our beekeeping team, or bee team up at Newcastle. And all of the bees that I talk about today and the experiments I talk about today came from these colonies. The presentation is going to be really quite simple. We're going to start out with what do we know, and then we're going to talk a little bit about progress so far. So the what do we know, those of you who came to my lecture yesterday will know that I love dipping into the really old literature. It's something that I very much enjoy doing. I think you can learn a lot from literature which hasn't been digitised, and so I'll be picking out some of my favourite papers on chronic bee paralysis, the disease, or chronic bee paralysis virus from the last 100 years or so. As part of that sort of sharing that past knowledge. And then I'll talk about some of the research outputs that we've found along the way in more recent years. So what do we know? What do we already know about chronic bee paralysis? Well, we know the symptoms, and you guys need to know the symptoms. Originally, when Bailey tried to characterise this disease, he had to try and pull all of these different diseases that were in the literature together to be caused by a single causative agent. And to do that, he talked about 2 different types of symptoms. We have trembling, shivering, and shaking. That's type 1 symptoms, and we'll see those on this video here. I want you to pick a bee, and I want you to watch a bee, and just watch those repeated movements of the bee, okay? Moving forwards and backwards, shivering, shaking the wings, very, very unusual. Quite often these bees won't respond to smoke very well, Okay, they don't always clear. They find themselves displaced on top bars. You squash them very easily because they don't move out the way. Some researchers back in the 1930s talked about the bees being stupid. Okay, they're just not doing what they should be doing. Okay, so that's type 1 symptoms. In our experience, as the disease progresses, later in the season, later in the disease epidemic, you start to see type 2 symptoms, which are where you see these hairless individuals, often shiny, greasy-looking individuals. Sometimes they look a little bit nibbled, the wings look nibbled, and those hairs have been removed by probably the sisters chewing at them and gnawing at them and trying to get them out of the colony. So they're the individual level symptoms, and in our experience, they tend to occur a little bit later. These individuals also will often be shaking. I don't like to see just a black robber-style bee being reported as chronic bee paralysis, because unless it's showing symptoms, for me, type 1 symptoms, for me, that's not quite good enough, okay? What happens to these bees that get infected? Well, all of those bees we saw shivering and shaking will be dead anywhere between 5 and 8 days. They will die. They'll be thrown out the front if the colony is strong enough to do so, or they'll remain on the high floor. And this can lead to these massive piles of dead bees that seem to appear very, very rapidly. Okay. When we say dead bees, there's another pile of dead bees. They're not really dead. Can you see the change in coloration here? Just here. These are not dead bees. If you go and look at them and you blow on them, they'll be paralyzed. They might start to twitch a tarsi or a leg or something. You might see that they're actually still alive if you go and blow on them, but they're actually paralyzed. So they can't prevent themselves being thrown out by their sisters. Okay, they haven't happened out there on their own. They haven't walked out. Actually, they've been thrown out. And there's a video here. This is a call out I got taken to, I'd say, would have been about June, I think, in 2022 in Yorkshire. Small-scale beekeeper, not a professional beekeeper, a couple of colonies, and this was one of them. You can see the amount of dead bees out the front. They're actually blowing around, so the pile's quite spread out, and then all of these bees on the bottom board as well. Let's have a look at the video as we scan over, and you'll see what I mean by this twitching. Okay? If you look carefully, particularly towards the end of this video, you start to see individuals, a lot of individuals, which are just twitching. Can you see? Yeah? There's this little twitches still going on. They're not dead, but they are paralyzed. Okay? So that probably hopefully gives you a little bit more idea about what the disease looks like. So a quick show of hands then. Having seen those symptoms, how many of you think you've seen chronic bee paralysis? Even more. Great. so we have educated you. Right, so that's loads of people. Great. So what's causing this? Well, I mentioned earlier it's chronic bee paralysis virus. What does that look like? Well, you'll not be able to see this in your apiaries. This is an old electron micrograph of the virus. And what's cool about this virus to me, as somebody who's interested in viruses and in pathogens, is that these particles are asymmetric. They're called anisymmetric. So they're lots of different shapes, lots of different sizes. And it's a very unusual virus. All the other viruses, pretty much, that infect honeybees, bar one or two, are these icosahedral, almost circular shapes. This one isn't, and this virus is particularly unusual because if we take the genetic sequence of the virus and we try to compare it with other things around the world that we found, not just in bees, but anywhere else in the world, this virus stands alone. It's a complete oddity in the world. There's nothing that's really that closely related to it, and so currently it's unclassified as a virus. I think that in itself is quite staggering that there's only one type of virus like this virus in the world, even though we've done all of this searching and we've done a fantastic job of adding information to various databases around the world. There's still nothing quite like this virus. It is a real oddity. So how does this virus move around? Lots of different ways. Okay, it could be fecal oral. I think that's a really important route of transmission. So this is effectively the virus getting into the digestive tract of a bee and managing to then infect that bee and cause symptoms in that bee. Okay, that's really, really super important. Bees do an awful lot of sharing of food. They do anal grooming. They groom each other. There's been some videos of bees grooming this week already. And so I think that's a really important route of transmission. Another important route of transmission is mechanical. So this virus is able to move from bee to bee by two bees rubbing up against each other. Okay, so that's a really important source of transmission when we start to think about how many bees there might be in a bee box in a colony. Transovarial. Now, there's a couple of papers out there which talk about this idea that the virus can be passed from the queen through her eggs to her offspring. We haven't seen very good evidence for that in the literature. It's really using some very sensitive techniques to detect tiny amounts of virus. And in general, when people look at brood, the levels of chronic bee paralysis virus are very low or absent. So I don't think it's a very important route of transmission. And then injection. And you might say, well, that's a bit odd, injecting, why would that happen? I guess you've got your varroamite, that might be injecting things as it goes around feeding. But actually, this virus isn't known to be transmitted by the varroamite. It's one of the few viruses that isn't. And injection is actually a way that many researchers over the years have tried to expose honeybees to this virus and then try to learn a little bit more about the epidemiology and understand the disease better by injecting. And you really don't need many virus particles to cause infection through injection. So you can imagine that all of these transmission routes are going on all the time in a colony, and what we were interested in trying to do was tease them apart and understand them better through the work that we did. So let's have a look at one of my favourite papers. This paper is from 1933 by a guy called Burnside. He did some extensive work on chronic bee paralysis. Some really, really groundbreaking work. His work is often overlooked a little bit in the modern literature, but he did some really, really cool stuff. And he was working at a research station, and somebody brought to him some colonies that were suffering from chronic bee paralysis. And so he wanted to do some experiments to understand better how the disease might be transmitted. So the disease colonies were headed by Italian queens, and that's important as I go through the evidence that he presented. First of all, what he did is he took whole frames of pollen and honey and he moved them into some carniolan colonies to see whether or not by moving the stores, the pollen and the honey, he could also move the disease. He got no disease. And he did this quite a few times, actually, over the years through the 1930s, but he got no disease. So interesting, right? So you can move whole frames. It's not like European foulbrood. You do this with European foulbrood, you'll move European foulbrood, without a doubt, but you don't move chronic bee paralysis as a disease. Okay, let's see what else he tried. He took 3 frames of brood from these diseased colonies and he put them into carniolan colonies to see whether or not that brood coming through and that whole movement of frames would move the disease. No disease. This is getting curious, right? So you can take whole sections of this colony and you can put them into other colonies and you will not move the disease. Okay? But you needed to do something with those frames of brood from the carniolan colony. So you put them back in the Stickitalians. And this is where you got the really interesting result. Because when you look at the carniolans, they're darker than the Ligurian, the Italians, which are very much lighter offspring. You could actually see that disease developed in both stocks. Both those that came from healthy and those that came from the diseased stocks. Both showed symptoms. So that tells us a really important piece of information in that this is an adult bee disease. Okay, this is a disease which is circulating within the adult population, and you can shift whole parts of the colony into new hives and you won't move the disease. So another one of my favourite papers was by Bailey in 1983, and he did some really cool work where he exposed some bees to chronic bee paralysis. You can see those ones here, okay? And he took them, those symptomatic bees, and he put them into pots with healthy bees, okay? And the pots he fiddled around with, and he kept the same ratio of diseased to healthy, and he just made them more dense, okay? So he put many more of these bees in another pot, and he wanted to monitor how many of these bees transmitted disease to how many of these bees. So it's a very simple transmission study that really just modifies the density of that population. So how did it go? Well, actually, in the less dense pot, 32% of those healthy bees developed chronic bee paralysis, the disease, and in the more dense part, 84% did. So there's very clearly a relationship here with density. So this concept of mechanical transmission, the virus moving from one bee to another could well be very important in this particular result. Alternative hosts, this is another one of my favourite papers. It's a really interesting paper, all these two are, because what they talk about are finding this virus replicating in other organisms, okay? And ants in particular have been found to carry very high levels. This is a picture of one of my colonies overwintering. I usually put a bit of candy on the top and then I watch as that candy goes down, and then I realise the bees are nearer the top and they might need a little bit more. Stops them running out of room and running out of food. But if you look here, every year I get these little ant colonies that form and they sit on the bag because that's the warm bit. Okay, that's where the heat goes. So we often find ants in our colonies, and it's just interesting to see that actually these ants, not only is the virus replicating in them, but it's replicating to thousands of times higher levels than you would find in a honeybee. So it's very clear to me this is not a honeybee virus, and I don't like the term honeybee virus because actually many of the viruses that are in honeybees and been found in honeybees are actually more generally insect viruses. So this is definitely an insect virus. Okay, I'll do it for time. Perfect. So let's talk through some knowledge gaps then. We're going to talk about some of the research that we wanted to try to conduct. These are some of the sorts of questions we're interested in trying to answer. Is chronic bee paralysis re-emerging in the UK? Question, right? How common is the virus? What are the disease risk factors? Are some bees more susceptible than others? Can pollen help prevent disease? Are dead bees a source of infection? Does the disease just appear? Are drones a source of infection? And which control options work? Now, if you stay with me to the end of this, this lecture, we'll hopefully run through and answer almost all of those questions, okay? So let's have a look at progress so far. The first one was about emergence, or more specifically re-emergence, because this disease has probably been at very high levels in the past, across England and Wales, and then it's dropped again, and then it's come up again, then it's dropped again, and we're in the process. The question here is, are we in the process of another re-emergence of this disease? And to answer this question, we got some information from BeeBase, which is where the bee unit inspectors put all of their information for the inspections that they carry out. And we took information from nearly 380,000 colony records over the years 2006 to 2017. It's nearly 80,000 visits. And we went looking for information. Now, in there, they're not there to record chronic bee paralysis. They're there to record the presence of European or American foulbrood. So it was a bit of a challenge, and we had to try to figure out a way around the fact that they don't have to record it. There is a box, actually, there is a checkbox, but they don't always check the box for chronic bee paralysis. So we went looking in the comments section. Okay, so there's a comments on there. Did you know there's a comments box when they're filling out your inspection? They write things on there, and maybe they write things like, "This is my favourite beekeeper," or "Absolutely the best lemon cake that I've ever had." Okay, but they write things on there that give an idea about how the colony's doing. And what we were interested in is picking up any signal whereby the bee inspector looked at that colony and felt it merited mentioning some of these symptoms. Okay. So maybe it was paralysis, trembling, shivering, shaking. You can look in the paper that I'll quote in a minute. It's got the full list of the things that we look for. And then what we did is we pulled out every individual record that had any of these keywords that might relate to chronic bee paralysis. And we read each one individually to make sure that it actually was referring to what we thought might be chronic bee paralysis with a description. And that's really important because what you don't want to pick out are records that perhaps say this is definitely not paralysis, okay? Which if you don't go back and eyeball the data, you might have done. So we did that, bit of due diligence on the data. Okay, what did we find? Well, in 2006, we found no cases in BeeBase that seemed to be recording the presence of chronic bee paralysis. But then we started to see a change over time. We started to see the number of cases that were being detected in BeeBase rise exponentially, and up it goes up to 2017. Okay, so we saw this increase in cases. And then we can see, if we look at these maps, we can see where those cases are. We can see that the intensity, these are sort of kernel smooth plots, okay? So you can see where those cases are. We can see these sort of more intensive outbreaks in different areas across different years. But it's got a bit of an unusual situation with it, because normally when you have a re-emerging disease or an emerging disease, you might expect a single point of infection, and then you might expect things spreading from that single point of infection. That's not what we have here. So we've got something which might be present at lots of these locations that's not always causing a problem at all of these locations. So it's an interesting pattern, but we can definitely say it's re-emerging. What about how common is the virus? Well, if we look at some of the literature, we can see how common the virus might be in places like France. Okay, we find in surveys there about 1 in 3 apiaries, something like that, will test positive for the virus, but only the disease in about 1.5% of apiaries. Okay, so the virus is far more common than the disease. How about in the UK? We found in the UK that chronic bee paralysis virus is in about 30% of colonies, but again, the disease is in about 1% of colonies. So this is very interesting. So what it means is the virus is far more common and out there in the landscape than it is actually able to cause disease, which starts to raise some questions as to why does it sometimes cause disease. And when it does cause disease, we can say that this is the log amount of chronic bee paralysis virus in different colonies that are either asymptomatic or symptomatic. We can see that we get a lot more virus in those that actually show symptoms. So this virus goes from being there to being there in massive quantities in maybe 1 in 30 of the apiaries that it's present in. Okay, so let's try and understand what the risk factors are around this particular disease. So one of the nice things about working with modelers and epidemiologists up at Newcastle is that we can have a look at these data and pull out a lot more information from data sets than we're able to before. And we could say things like, what are the apiary level risk factors? If we take all of the cases of chronic bee paralysis from 2007 to 2017, what things do they have in common? Well, the first thing they have in common is that each year there's a 25% increase in the chance of any apiary testing positive. So that's quite a lot when you think that we've been looking over those years. What else can we say? We can say professional beekeepers, that's those with 40 or more colonies, they're 1.5 times, so they're 50% more likely to catch or show symptoms of chronic bee paralysis. It probably isn't catching, that's the wrong word. Okay, but show symptoms. So there's something in that. Importation, those that imported within the last 2 years had about an 80% increase in their chance of showing their apiaries showing chronic bee paralysis. Okay, so we start to get a few clues here, don't we, as to what might be actually causing this virus to go from being present to causing a problem. We also brought in a load of data. This is when we were in the EU, we used to get this kind of information. These are lots of different imports from lots of different places. And we wanted to investigate whether or not there was a regional risk factor for importation from particular places. Okay, so we're taking that importation result from the previous slide and we're going to pick at it a little bit and see if we can find out any more information. And so we looked to see if there was any increased risk in each region based on whether or not we had these imports from different places. And what we found was this really small increase. So it's Danish imports and it's a 3.5 additional cases in that county per 1,000 imports. Okay? So I want you to think about that as a number. It's a very, very small number, but we were able to see a significant increase based on Danish imports. But that increase was really, really small. Okay? When you're dealing with big data sets, you can get significant outcomes which can be very, very, very small and nuanced. So we wanted to find out, we didn't want everyone running around and saying, let's never import from Denmark again, these things are filthy. They're all going to die of chronic bee paralysis, which there's risk of. And there's also, when you start talking about imports, there's a lot of people that are very interested in trying to promote local bees. And of course, they were very interested in saying, great, look, this is great evidence to say don't import. It isn't great evidence for that, actually. It's just trying to allocate risk in a very subtle way to counties based on the features within those counties. So we wanted to try to understand this a little bit more, and we tried to pull it apart as scientists. We came up with a couple of hypotheses H1 and H2, you know, got very scientific about it. The first H1 is imported honeybees are bringing chronic bee paralysis virus with them, right? That must be how it's getting in here. The apiary is clean, and then you bring the import in, and then the apiary is dirty, and then you get the disease, okay? So that's a reasonable hypothesis, right? A second reasonable hypothesis is that the imported honeybees are coming in completely clean, but they're more susceptible to chronic bee paralysis because of something, okay? So there's an endemic virus that's here that they're not used to seeing, they're not used to coping with, and they have some features about them as imported bees which effectively increase their susceptibility. So we wanted to try to separate this, and I'd say Brexit got in the way a little bit, because Nicola Simcock, who's one of the postdocs up at the lab, she tried to set up an experiment and did a good job of setting up an experiment. We wanted to do this for 2 years, but we were only able to do it for 1 in the end because of legislative changes around what happened to the attending workers. But what she did is she contacted some bee farmers, and those bee farmers told us that they were going to import, and we ended up looking at importers from Denmark in particular, and we got 5 importers, and what they agreed to do was to provide an adult bee sample from their colony that we were to test for chronic bee paralysis virus before they imported. And then we took the attending workers from the specific queen that went into that colony, and we tested those for chronic bee paralysis virus. And then we waited over some time, over 1 or 2 seasons actually, sometimes it was within the same season and sometimes the next season, to test those bees and to see whether or not we saw symptoms arising in those colonies. Okay, so we've got 32 colonies in the study, 32 queens in the study, and 5 importers. Okay, first, every single apiary, or one colony, at least one colony from every importer, had chronic bee paralysis virus in it. Okay, so this is interesting, right? So we know the virus is present in all of the apiaries where we were importing these queens into before the queens arrived. What about the attending workers? Well, none of the attending workers, and we tested all of them, actually contained any chronic bee paralysis virus. So it seemed reasonable that the queen then wouldn't have the virus in her, given the fact that she was fed and housed next to these attending workers. That's an assumption that we made. What about afterwards? Well, actually, we had one colony develop with chronic bee paralysis, the disease, afterwards. So this was quite a nice experiment because it helps us to perhaps point the finger that the genotypes we're importing are being exposed to chronic bee paralysis virus when they come into the country and then developing disease as a result. Okay? Okay, next bit. Time, 1/2 Okay. Are some bees more susceptible than others? And so we've got different people on here. We've got Nicola again, and then we've got Theodora here, who's a PhD student, who's going to have her Viva examination next month, so she's nearly finished. And what we're interested in answering here is, within a colony, are all the ages of honeybees equally susceptible? We've established that it's an adult bee disease, but is there a window of opportunity for that virus to cause disease within these bees, dependent on their age? So we took emerging bees directly from a comb, and we effectively tried to simulate fecal oral infection. Okay, so this isn't quite how they probably imagined it when they were born, when they were closed, but they got whipped up and they got taken off into a lab, and Theodora very, very gently picked them up by their wings after starving them for an hour or so, making them nice and hungry, and fed them a load of chronic bee paralysis virus. Okay? So this was their life. They ended up being fed this. And we're trying to simulate what might happen. So when the bees emerge, they do get fed by those around them, and they often go and seek a bit of pollen and a bit of honey. And so we're trying to simulate that early exposure. So what did we test? We put them into pots, effectively. So we put them back into pots. We call them cages in science, but they're little pots. And we fed them some syrup, and we watched them over time, and we tried to find out whether or not they died or didn't die. Okay? And we've got different ages here, but we did this experiment 2 or 3 times, and we've done this all the way up to 48-hour-old bees. After then, they get a little bit flightier, a bit more difficult to handle. But yeah, so we've got enclosed bees, if you like, straight off the frame. We've got those that are 3 to 7 hours old, 24 hours old, and we've done some older ones too, but I'm not going to show you those data today. And we compared those to 3 different coloured lines, which are effectively bees that were the same ages, but actually fed some saline, just what we would have mixed the virus into, and we fed those just standard salt solution, effectively with a little bit of sugar. So what did we find? Well, first of all, we were really pleased because we started to see symptoms in these bees that we had actually exposed. This is not the best video, it's a bit jerky, but you can hopefully see some tongue lolling there, tongues hanging down, we've got some repeated movements, and that individual is showing symptoms of paralysis, and when we looked into those samples, we found the virus at very high levels. So we know that we were getting the virus in there. How does the mortality look? Well, we've got 2 different patterns here. We've got those that just keep living, and they'd keep living until about 30 days, something like that. And then we've got those that die very rapidly between days 5 and 7, 5 and 8, something like that. And then they flatten out and they live until about 30 days. So there's 2 patterns to the mortality curve. Okay? The pattern at the top would be those bees that were exposed to chronic bee paralysis at 24 hours and any of the controls. They all behaved in exactly the same way. Those ones below are bees that are younger, between 3 and 7 hours old, or are closed. Those bees actually suffered from chronic bee paralysis, and they tested very, very highly positive for chronic bee paralysis virus. I'm not showing you those results today, but they did. Okay, so this is really interesting. As soon as a bee gets to 24 hours old, it is not susceptible to chronic bee paralysis virus by fecal oral route, at least not in our pot studies. What about, can pollen help prevent disease? Well, pollen is something that we don't tend to use very often in pot studies in the labs. Okay, so to add it is a little bit more unusual. It's one of those things that's much more difficult to control in an experimental situation. So a lot of the pot studies you see in papers don't include pollen, but we all know pollen increases survival. But we wondered whether or not it might help these bees, these adult bees, with coping with that oral exposure to chronic bee paralysis. So we just basically got some fresh pollen, had been stored down, so this is bee bread effectively, and we squidged it into these little bits of end of tips and weighed them, and we put them into the pots, and we let some bees have access to these that had been exposed to chronic bee and some not, and wanted to compare the mortality. And what we found was, same thing, same exposure, Theodora working away. We found that if in this experiment, that all of them died actually. All of the ones that weren't given pollen died, but only half of them that were given pollen died. And this looks like it's not a great thing either, because only half of them survived, but that's a lot better than none of them surviving in this particular experiment. So we do see sometimes some variation between the number of bees that die when they're exposed to chronic bee paralysis between experiments. That often happens. And in this experiment, we halved the death due to chronic bee paralysis virus, which we thought was pretty good. So the next one is, are dead bees an important source of infection? It's been shown in pot studies before that they can transmit the virus, but it's not really been quantified. We wanted to take that one step further. Now, this is Megan. She's one of our master's students, and she ran this experiment last summer. We took some dead bees from outside a colony that had disease symptoms, and we wanted to find out how frequently they might be passing on disease to their sisters in a confined environment. Same thing, we take these dead bees, and I've gone to the effort putting crosses on their eyes, look, they're obviously dead. It's a little bit like a cartoon, doesn't it? But so these ones are dead, but they were infected with virus when they died. We checked that before we used them. And then these ones are bees that were healthy until we killed them. So writing the paper up is quite tricky, because you want to say healthy dead bees, but actually no dead bees very healthy. So we settled on corpses from healthy colonies instead. But yeah, so we've got those in there too. And we're looking to see how often the disease is passed to these individuals compared to the mortality in this group here. This is one of our cages. We've had these made. They all effectively mimic bee space, so it's trying to get that kind of individual seam of bees. We can modify the area here as well. And what did Megan find? What she found with the corpses of healthy bees, she found that this was sort of the response over about 3 weeks, okay? You can see that we have a few dead bees coming in here in purple, and at the top here we have asymptomatic, and those bees are still alive, okay? We did measure the symptoms. I'm not going to show those to you today. What do we find in those with corpses? Well, we can see that pink area rockets up, okay? And then there's a little plateau, and it rockets up, and it plateaus, and it rockets, and it keeps going until all those bees are dead within just over 2 weeks. So actually that's quite a significant difference in those two results. When we tested the bees in the pink area here, all but 4 across all of our pots were positive for chronic bee paralysis virus. So these dead bees are a really important source of infection. They can really rip through, the virus can really rip through sisters that are ending up being kept next to dead bees. So another question then was, does the disease just appear? And this is some work we did with Philippa Holder. She's a postdoc who was with us for a few years. And we went out and we contacted some bee farmers that had problems with chronic bee paralysis in previous years. And we asked if we could set up nice and early in the season in 3 apiaries that had had a repeated history of chronic bee paralysis, the disease. You can see us here setting up lots of monitors. Did a really good job. We put dead bee traps outside. This is another site. You can see the dead bee traps here outside the hives. And then another site disappears, I think, behind Philippa. But you can see what we were working with. We're trying to set up 5 colonies in each of these 3 apiaries, and we're going to go back, and we're going to go back, and we're going to go back every 2 weeks. We did a 600-odd mile round trip to visit all these 3, collect data, collect samples, and wait for the disease to occur. Now, this is not easy to do, because the chances are the disease won't occur at all. And it was really hard. This is the results for the first 5 visits up to May, look. It was really hard to encourage Philippa to jump in the car for a 600-mile round trip after we'd done all of these and seen no disease at all. But thankfully, she stuck with it, and we went and did a few more, and we started to see the disease occurring. And we got the disease in the end in 6 of the 15 colonies, which is no mean feat for chronic bee paralysis, actually. And we saw a really interesting pattern. We saw that if you wanted, the S's represent symptoms that we could see. So this is what Philippa and I could see in the colonies. The trap tells us that we're seeing a buildup of dead bees in the trap, but we can't see any symptoms in the colony. Okay, so the trap's a really good way of getting a jump on any symptoms you might see in the colony by 2 weeks. The beekeepers that we were visiting, they said that they would only see the symptoms here. So they kind of didn't, they could see the symptoms when we pointed them out here, but they said they wouldn't have noticed them. But they did say they'd noticed them 2 weeks later. This is 4 weeks after the dead bee trap is recording dead bees, then beekeepers would likely see the results of those bees dying. And these are very, very experienced beekeepers that have a lot of experience with this disease. They're certainly not newbies when it comes to this disease at all. So this was interesting. So we're thinking to ourselves, does it just appear? Well, actually, no, it's already causing a build-up of dead bees 4 weeks before. But when we went back and we looked at those bees we've been collecting from the dead bee trap or from inside the hive for the weeks and months before, we found a massive load of virus in dead bees from 3 months before we saw symptoms. So right back in March, we were seeing bees dying of chronic bee paralysis virus. And so I think we need to rethink, actually, how this disease works. This idea that you go to a colony 1 week and it's fine, and then you do an inspection 10 days later, and it's got thousands and thousands of dead bees in it, and where did that disease come from? Actually, that disease has been burning through and slow burning, and it's been slowly increasing over time. And in some colonies it takes hold, in other colonies it doesn't. And that's a different research question. Okay. So what about drones? Why am I talking about drones? Well, those of you who've seen symptoms, did you see symptoms in drones at all? Any symptoms in drones when you saw it in your bees? Do you remember? Now, hands up if you think there were. Okay, no. Okay, well, one of the things that we found striking was symptoms in drones when we were looking around. And I suppose we're a little bit curious with our minds, but we did notice that every colony we found with chronic bee paralysis had some drones that seemed to be struggling with chronic bee paralysis. Okay, we tested them, they were positive. These are some of the results from the dead bees over time. We can see what the circles on the outside mean. This is a mount of virus, log amount of virus, so this is nothing and this is really lots. And then the circles that are yellow are when the colony was asymptomatic, and this is when it turns to be symptomatic, and this is the level of virus in those dead bees over time. What about drones? Okay, hello. Drones, we can also find that they contain virus. Okay, so not only are they showing symptoms to us anyway now in the apiaries we visited, but they also carry high levels of virus, they can do, and also we wanted to find out if they could fly. Okay, so we took some of these sorts of individuals. You can see a bee here on the left, really struggling with chronic bee paralysis. She's shiny, she's shaky, she's got repeated movements. Okay, and we collected some drones as well. We looked at some drones. You can see some drones in here. A lot of those drones were symptomatic as well. And we actually got them when we threw them in the air. Okay, it seems like an odd thing to do, doesn't it? But we're in an apiary that's got quite a lot of chronic bee in it, and we thought we'd take some of these individuals and throw them in the air and see whether they had early symptoms, which is just twitching, or late symptoms, which includes the shininess. Were they able to fly? Okay, were they able to fly around and seemingly orientate back to the colony that we'd actually got them from? And you can see here that actually most of the workers, really quite surprisingly, were able to. About 50% of those with late symptoms weren't able to. But when we looked at the drones, and I wish we'd done more drones, but we didn't, but all 50 drones, no matter how sick they looked, just went zzz and went and orientated back to the colony, which was really quite surprising. So are they a source of infection? I think they are a source of infection. How important they are for transmission within a colony, you'll have to wait for future instalments. Okay? So, next question, do any control options actually work? Okay, has anyone heard of the sort of idea of shaking bees out which have got chronic bee paralysis? So you cage the queen and then you go about 50 feet away and you shake the bees out and then only the healthy ones return. Has anyone heard of that one? Yeah, quite a few. Okay, so whenever I try and give a talk on this or talk to bee farmers or people phone me up, I ask them if they've tried this method. And I asked them if it was a success, and I wish I'd been writing the numbers down, but I haven't. And about half the time it succeeds, and about half the time the colony falls over. And if I tell you that 40% of the colonies that show colony-level symptoms actually die, then I'm not sure there's a lot of evidence for this working. And actually, because honeybees can actually develop the symptoms over a number of days from becoming exposed to the virus, we've seen that on some of our plots earlier, and because some of those bees that are showing symptoms can still fly, I'm not sure that this really works out, but it'd be nice to see some good hard data on it. In the literature, this is my favourite one, and I did look really long and hard. Loads of people recommend replacing the queen. There's no data at all in the literature that backs this up for evidence. There's no kind of paired study that says we replace the queen in half the colonies with chronic bee and not the other half. But it's in almost every book as advice to beekeepers to do. Not saying it doesn't work, it might be amazing, but there's no evidence that it works, okay? There's another one is to remove the floor, okay? Removing the floor and putting a super underneath and letting those this is managing the dead bees basically, which is what we're doing. I put this up there. But apparently this is another method that people are trying. Anyone heard of this one, where you remove the floors? Yeah. Yeah, NBU do this, yeah, they do recommend this. Again, we haven't collected any evidence that suggests that this is going to be good, because we haven't kept with the paired control, right? Now, it's very difficult because you've got your bees and they're dying in front of you, but we have to try to find out what works, which means that we've got to take some of those colonies and just leave them, okay? Just leave them like we were doing a few years ago when the answer to this was actually there's nothing you can do. We need to leave a few, and we need to start to try to get evidence. We've got evidence-based control policies. And that's what we want to do this year. The work is delayed by 2 years because of COVID and the problems that we had in the lab with COVID. We were using all the same reagents as the testing labs that were testing for COVID. So quite rightly, we got bumped right down all the way to the bottom of the list. And we ran the lab probably for about 18 months with very little ability to do anything, actually. So These trials are delayed, but what we're really interested in is trying some preventative trials that might not work, and some curative trials, so when you've actually got the disease symptoms, which still might not work, but they might work. So at this point, I say we need you. There's loads of hands went up that see it. I don't know how often you see it. Last question then, how many of you see it regularly in your bees? Right, not so many, right? So the people that just put their hands up at the end, if you'd like to come and see me, and you want to be involved in this work, then that would be great. But yeah, so if you do see it out there and you see it in more than one colony, and there's a couple of colonies that we might be able to do something with and nothing with, then get in touch, okay? So let's go through these answers. Is chronic bee paralysis re-emerging in the UK? Yes, it is. How common is the virus? Well, it's actually very common. It's very different from managing something like European foulbrood, where the causative agent is actually pretty rare outside those areas where you have disease problems. What are the disease risk factors? Well, we know that it's increasing in time, professional operations, and certain imports increase risk. Are some bees more susceptible than others? Yes, they are. Young bees are very susceptible. Bees more than 24 hours old, you can't get it in them. Can pollen help prevent disease? Yes, it can. Are dead bees an important source of infection? They very much are. And does the disease just appear? No, it builds up slowly over time and sometimes takes over the hive and sometimes doesn't. We don't know why. Are drones a source of infection? Yes, they are. How important they are in transmission? We don't know. And do any control options work? We don't know. Okay, so it's such a shame to get to the end of that and actually say we don't know with the last one. But hey-ho, that's science, and we'll keep going. Okay, so thanks to everybody on here for their contributions, funders as well. We're always trying to get a little bit more in for these students. So if you did want to take a picture of that, will take you through to a little donation site where you can add a little bit to the pot to help with some of these experiments. Thank you for your attention, and I'm very happy to take any questions. Yeah, so the question, I'll try and remember to repeat the question. The question was, is there anything seasonal about the disease? And it used to be known as May disease, which meant that it was often seen in May, and then you'd see this drop in occurrence of the disease. But actually, more recently, we see it rise up in May, so we can see it getting more common in the data up until about May, and then it flattens off in June, and it stays high through the year up until about September, and then it drops away again, okay? So yeah, it tends to not have the same seasonality that it used to be reported as having, if that helps answer the question.
Hello. Could you speak a bit about the biology of the virus, why it causes shaking and why it causes a darkening of the bee itself, and possibly also discuss, does it affect queens at all as well?
Yeah, lots of questions in there. The last one about queens I'll start with. It does affect queens. Queens less frequently go down with it, interestingly, but they can go down with it. And there's a few good studies in the literature which look specifically at the susceptibility of queens, so you can look those up. The question about darkening, I'm not sure about darkening, whether or not that's a response to the viral infection or whether that's a response to having the hairs removed, because quite often bees have a much darker cuticle than they do the hairs. And I'm not sure whether it's one or the other, if that helps.
You mentioned that dead bees are a source of the virus and the infection. Is that time-bound? Does that disappear after a period?
Yeah, and we're not sure, because what we didn't do... did everyone hear the question? Yeah, okay. I'm not sure, because what we didn't do was effectively put bees in of different ages. We put bees in that were all young. So I'm not sure, and I'm not sure how long those bees will remain infectious, if that's the question.
Yes.
We don't know that, but we are checking that this year. So what we've done this year is we're taking some debris from out the front of a colony that had disease last year, and we're going to try and do the same exposure assay and see whether or not it's still infectious. Yeah, so that could carry from season to season. Was that the question? Yeah.
Yeah. I've got a colony that died last year, and we've got dead bees in amongst the debris on the ground, and it's very difficult to remove all of them.
It is. Yeah, it is, and it's amazing how long they stay there, isn't it?
Yes.
Mm. So we'll be able to answer that next year. hopefully.
I'm just wondering, another thing I've heard of as a possible means of preventing it or stopping the progression is because it's associated with congestion of the hive, so the mechanical spread of the disease. If you give the colony more space, then it's less likely to transmit and cause problems. But whether it's taking brood frames out or bees out or giving them extra supers or giving them another brood box. I don't know.
It's a good point. Thanks for raising that. That's something that sits very nicely with Bailey's results, doesn't it? Where you get this higher rate of transmission when the bees are congested. We tried to do some work on that, but we didn't get infection into those pots that I showed you that changed density. That's exactly why we had those, so that we could change the density of our experimental pots, but we didn't get infection in those experiments, unfortunately. People have talked about that, increasing space and giving them more room, but I haven't seen any data where people went, more room for you, no room for you, let's see what the outcomes are. And I think that's where we need to get to. We really need to try as a community, because there's enough chronic bee out there, but as a community come together and say, right, we're going to actually work some sort of collaborative experiment to try to improve our understanding of treatment success and treatment efficacy.
Two questions. The change in the shape of the abdomen, what's that caused? How does that come about? And the second one is, do you think a change from Hoffman spacing to narrow spacing might also assist in reducing congestion?
Two good questions, of which I'm going to say I don't know. So, I don't know. Norman, do you know what causes the distended abdomens? Is it because they're not having cleansing flights, or?
Yeah, I don't know. It was always described as a symptom of Isle of Wight disease, and these bees with a distended abdomen. But whether it's that the virus is in the gut somehow, or as you say, they just haven't been out of the hive. I'm sure Bailey sort of dismissed that as, I think, so the very first study of Isle of Wight disease by Ims, who was the very eminent entomologist but didn't know anything about bees, he wrote this paper describing the symptoms of Isle of Wight disease, and then Bailey said, well, he's just describing bees that haven't had a cleansing flight. So
cleansing flight was my best guess, and Norman's as well, I think. Yeah, John?
Thank you, Giles. I was really just wanted to know, we've had the introduction of a potential vaccine against American foulbrood by transgenerational immune priming. Is this virus potentially a candidate that would benefit from that?
I think potentially. It depends whether or not viruses and virus fragments end up going through the same process for transgenerational immune priming, and I'm not sure if that's been shown, because it tends to be bacterial pathogens that have been tried for exploiting that route of passing some form of immunity to offspring through the queen. I've not seen it done yet with viruses, and so I'm not sure if it's possible to do that with viruses. There's actually, I guess I'm not convinced yet that you can do it with bacteria either, but I'm looking forward to being convinced. Yeah. So the question was, does it lay dormant within individual bees, right? And the answer to that is probably not, because we've got methods that will detect incredibly low levels of virus, and we can get negative bees. Okay, so it would have to be below just a couple of copies of virus, which is unlikely if it's going to be dormant. And you've got to remember as well that the only bee that's hanging around for a very long time is the queen. So the rest of the population's kind of completely changing over time. Okay.
If you do a shook swarm, you're then getting rid of, you'll have no young bees for 3 weeks, won't you? So the virus will still be there to pass on to the young bees when they actually emerge.
Yeah, I mean, are you saying we should do a shook swarm, or that we shouldn't do a shook swarm?
Well, in doing a shook swarm, yeah, you then will have no young bees for 3 weeks, will you?
There won't be any young bees for 3 weeks with a shook swarm, no.
No, so there's still enough virus left in there to actually Has
well,
it gone to the young bees that then start to emerge?
it depends how many of those bees that have it die. And I don't know what that question, the answer to that question. Yeah. I don't know how many of that population would still be alive after 3 weeks. The ones that get very high levels of virus, they won't be alive. But then if there's no bees that are healthy, I don't know if that would work. There's a few ideas we're kicking around which do involve colony manipulations, but that's not one of them. Yeah.
It sounds like what you're suggesting, Giles, is...
Wave at me.
Sorry. So I can take the brood, if it's sealed, out of a large colony with a queen that's prolific. I can put it in a split that I've created from another colony that was healthy. At least I'd get the brood and I'd keep the prolific queen. And what I really should be doing is, on prolific colonies, try and find the symptoms as early as you can. So that's kind of practical that we could all be trying now.
Yeah, definitely. Definitely familiarise yourself with the symptoms and try and be familiar enough with the symptoms that you can spot it before it's causing thousands of dead bees, because it's all about that timing of intervention that we might be able to do something that's successful. The idea about taking the capped bees away with the queen is exactly the kind of colony manipulation we're talking about trying, okay? Because we feel that there's a continual stream of susceptible individuals coming into that colony, and if we take them away from the source of infection, which we feel is the adult bees, then and the dead bees, then we think that that's a good chance of perhaps halting that B2B transmission. So if anyone's interested in trying these things, then do get in touch. Try not to do what beekeepers keep doing, and they have been doing since I started working on this, which is phoning me up and saying, I had a go at this last year, and it did work or it didn't work, because I can't do anything about that. It doesn't improve our knowledge or collective understanding. So if you are thinking about trying something, get in touch, and we'll try and include it in a structured way into an experiment. Okay? Yeah.
I have a practical question. What do you suggest a beekeeper should do if you had the situation we had last year when we had heaps of dead bees, first of all at one end of our apiary, and within a few days, all 12, 14 had heaps of dead bees. We didn't know what to do, so we swept them up with the dustpan and brush, took them home and burnt them, and waited. We lost most of our queens, they subsided, all the colonies survived, and we have our fingers crossed for this year. But what would your advice be to people who, when they first see the disease, and
if they first see the disease, I'd like them to contact me so we can include them in an experiment, right? But you're dead right. Clearing away the dead bees, I think, is important, because we've all seen bees coming back to the colony and having a rest. And they have a rest on whatever's near the entrance, don't they? And if you've got a big pile of manky dead bees near the entrance, you're definitely going to get some contact there. And we know that the virus will, those dead bees don't need to effectively try and share food with, but they just have to be contact, and then that's probably enough to keep it going. So tidying away the dead bees is the right thing to do. But over and above that, rather than giving out advice, I really want people to get in touch. And I want people to include their bees as part of a wider experiment. Yeah.
So a bit of a follow-on question, actually. How susceptible are neighbouring colonies in the same apiary?
Say again?
How susceptible are neighbouring colonies in the same apiary?
Okay, so how susceptible are neighbouring colonies in the same apiary? So my question would come back to you is, are all the entrances facing the same way, and do you get a lot of drift? If you get less drift, the risk is lowered. So you are going to get bees leaving a hive with chronic bee to forage and come back, and bees aren't very good at finding the right colony. We know that. We've done genetic testing which has found anything up to 66% of the adult bees in the colony were not born of the queen in that colony. Okay, so they're very good at mixing. Remember, they're single point foragers, and a single point would normally be in nature one nest in a tree somewhere. They're very good with, but when they turn up and there's a row of 15 boxes, they'll go in the nearest one, probably, which is why you get things at the end, a big colony at the end. It's always, if you ask beekeepers, it's always a colony at the end of their apiary which is their most productive. Okay, it's because they've been foraging that way and coming back to that one. One of the tricks, I think, with some bee farmers is if they're going to go to the heather, they're going to see where people have got their bees and put their bees in between the other bees and the heather, and that way the bees come back a bit closer. So I do think that you can have risk from one colony to the next, and reducing drifting is a good way of managing that risk, because the viral risk is likely to be in the adult population. Okay? Thank you. Yeah.
So just a massive thanks to Giles. I think we could have stayed here for a long time. I could see lots of hands going up, so sorry we didn't get to answer all the questions. So thanks to Giles. And also I want to give a shout out to Bucks County Beekeeping Association. I don't know if we have a representative here. Thank you for your sponsorship. Thank you very much.